So I'd like to welcome you to the annual George Corey Lecture at the NIH. It's my great pleasure today to actually introduce two outstanding NIH researchers. The first, of course, is George Corey, for whom this lecture is named and who was an incredible scientist at the NIH. Unfortunately died 25 years ago from lymphoma. And we've had this lecture virtually every year since then in his honor. Anyone who ever met George knew that he was going to be successful. I noticed Marilyn Corey in the audience, and she will assure us that that is the case. He was an honors graduate of Princeton, an honors graduate of Harvard Medical School, and then at the ripe old age of 33 became chief of the NCI virus tumor section at NIH and NCI. In his all too short career at the NIH, George managed to contribute in an incredible way to science here, publishing over 140 original research papers and earning membership in the National Academy of Sciences, which was awarded just a few months before his death. His primary interest was in regulation of gene expression in mammalian cells. We all know about the importance of sequences we call enhancers, but probably some people in the audience don't realize that the term really was, the term and the concept was invented by George Corey, and that is a lasting legacy for which we are all rather grateful. He made many seminal contributions to the field of virology. There wasn't very much known about how viruses caused cancer when he started his work at NIH in the 1970s. And he was among the first people to realize the way in which small viruses, such as SV40, were able to cause cancer by turning on gene expression and subverting normal growth mechanisms present in mammalian cells. His scientific prowess was only matched by his incredible mentoring. In that short period of time, a huge number of really successful scientists passed through his laboratory, and each one of them benefited from his individual care and attention. And many of us who were just friends and colleagues also benefited from his sage advice, his wisdom. He really loved science. He loved to go to seminars and would get deeply engaged in any scientific discussion on a whole variety of issues. He's the kind of person who really added pizzazz to the NIH intramural program, and we really, really miss him. So in his honor, we've created the George Corey Lecture Series. It's one of our Wednesday afternoon lectures. And we expect that every year a leading investigator, especially people who've been associated or had been associated with George in his career, would be one of the speakers. I should point out that there is another George Corey Memorial Lecture Series at the Wistar Institute. And I had the privilege last year to actually give that lecture, for which I was deeply honored. And it made me appreciate that George's impact extended way beyond the NIH. There were people in many, many institutions, his undergraduate institution at Princeton, the Wistar Institute, which he was engaged in over the years, who remember him extremely fondly and wanted also to memorialize him with a lecture series. So the second outstanding NIH intramural scientist I'll talk about today is our speaker. That's Jeang, one of these, quote, young scientists whom George Corey helped train. You need a little bit of backstory here. Every year, T helps choose a speaker for the Corey Lecture. These have been stellar presentations, as you know. Diane Griffin from Hopkins, Jeremy Nathans, also from Hopkins, Frank Rasher from Penn, just in the last 3 years. So T has done a terrific job, and with John Brady, who died unfortunately at an early age a few years ago, they really ran this lecture series for many years. And so this year, as I was fretting over who would be the best speaker, I had an epiphany, and what struck me was that T himself, who is a really truly outstanding scientist, would be a terrific speaker in this series. We invited him. He dithered a little bit about whether he really wanted to do it, but he did agree, and we're delighted to have him here today. Well, so T, many of you know, is a real dynamo. He's been a scientist at NIAID for 25 years, where he's now chief of the molecular virology section of the Laboratory of Molecular Microbiology. He's published over 250 scientific papers and has edited 6 books. He's the editor-in-chief of Retrovirology, an editor of Cell and Bioscience, and an associate editor for Cancer Research. He also serves on editorial boards for the JBC and the Journal of Virology. He's also president of the Society of Chinese Bioscientists in America, and in that capacity has been outspoken in his desire for increased representation in leadership positions for Asian American scientists. And he really has been very compelling in that position and hopefully will be extremely successful in advancing careers of very people who really deserve recognition. So some of his more recent work includes findings on genome-wide characterization of 252 human host cell factors and Jurkat T cells that are contributory to HIV-1 replication, and characterization of several oncogenic microRNAs and their contributions to HCLV-1 oncogenesis. And of course, this is recent, and the list of his contributions is almost endless. He was an undergraduate at MIT and got his MD and PhD from Hopkins University School of Medicine. So let's welcome Jeang, whose talk today is entitled Nuclear Damage and Aneuploidy, HTLV-1 and Leukemogenesis. Thank you.
Thank you very much, Michael. And it's indeed a great honor to be here to both give the lecture and also to celebrate George Corey and his contributions to science. As Michael mentioned, I was one of George's, I think, last postdoctoral fellows. And I'm really indebted to George in the sense that I learned a lot about science from him, and perhaps even more about being a human being and how to sort of conduct and teach science. So some of you have visited my office over the years, and I think you have always noticed, and especially new postdoctoral fellows who come in the lab, you've always noticed this picture hanging in my office, and it's been there for the last 25 years. And some people wonder, who is this person? And this is actually the picture that George gave to me a few weeks before he passed away. And it certainly is a picture that I've treasured and have hung it in my office for the last 25 years. Now, I would like to share a couple of other pictures with you. These are pictures of George and, a little bit earlier, before when he sort of started looking sort of wane from the disease. And so this is a very nice picture of George and Marilyn, and these are pictures of a party that we had at George and Marilyn's house during the period when George was doing well in his remission. And you can see some individuals, I think some of them are still here at the NIH. This is Lionel Feigenbaum, who is over there at NCI Frederick. I believe this is Jeff Green. It's remarkable how some of these people sort of have changed, and they look so much better when they were younger. Some of you know Kamal Khalili, who is now at Jefferson. This was Kamal when he was younger. This was, of course, George, and this is actually an image of me some 30 years ago. And this is Lou Lehman. And this lovely 20-some-year-old girl sitting there is actually my wife, Diane, who also came to the party. And here are a couple of pictures. Here's a picture of George with Nadia Rosenthal, which I know many of you are friends with. And Nadia was also here at the NIH many years ago. So these are some of the sort of off-hour good times that we had with George. And George was a terrific, not only a terrific scientist, but I think also a terrific mentor and a teacher to us. So with that then, shortly after George passed away, I had the desire that, because I felt that I had benefited so much from George, that I should do something to try to remember George. And so in 1994, with the help of Alan Rapson and Ruth Kirstein at the time, we started the George Corey Lecture. So this was the very first poster of the very first lecturer, Arnie Levine, who was a great friend of George's, who came and talked about P53 tumor suppressor, a topic that's very near and close near and dear to George's heart. And as you can see from those early days when we sort of were on sort of a low budget, we actually ended up having, you may not be able to read these names very well, but these were a number of George's postdoctoral fellows who personally contributed money to sort of bootstrap this very first lecture, including Joe Jeang, who I know is in the audience today. And so you can appreciate that back in those days when you first started a lecture with a low budget, that you sort of print the program and the announcement on this sort of brown bag paper. So that's really indicating that this is sort of the early days. But then by and by, we sort of got lucky, because when Michael became the intramural director, we were able to have the lecture become part of the Wednesday afternoon lecture series. And then you can see that we got much nicer poster designs and much better framing, and got a number of additional luminaries come to give the lecture. And indeed, over the 18-year history of the George Corey Lecture, you can see we've really had a number of all-stars that have come. These are friends, colleagues, students of George's that have come in and really have presented wonderful scientific talks, and also in many cases really personal stories about George. And so here's a picture of my friend and the former George Corey lecturer in 2009, Frank Rauscher, and this is a very nice picture of George that he showed during his lecture. Now, I want to say that with every successful lecture series, there is always a community of individuals. It certainly is not me, who we owe credit to. And in the case of the George Corey Lecture, there are really a number of individuals that we should credit, which is that initially, during the early days, Alan Rapson and Ruth Kirstein were very instrumental in helping me set up the lecture. And of course, over the course, we've always had very wonderful support from Marilyn. And we've also had Susan, Michael, and Ira sort of form the community of individuals who have really helped us get the lecture going and continuing the lecture in a high-quality way. We've also had Bernie, Taz, and Bruce, and Doug, and Warren Leonard, and Malcolm Martin. And so these are the individuals that we owe credit to over the last 18 years for continuing this lecture and really maintaining the high quality of the lecture. But out of all these people, the one person that I really want to acknowledge, perhaps in an outstanding way, is of course my friend and former colleague in George's lab, John Brady. John, as Michael mentioned, passed away a few years ago. And during the course of those 18 years, there was a period of time when I was actively negotiating for a position elsewhere. And during that time, John stepped in for several years because I was concerned that I needed someone that could really continue and do the George Corey Lecture in a really efficient and outstanding fashion. And so there were a large number of years there in the middle where John stepped in and did the bulk of the work, making sure that the lecture was done in a really outstanding fashion. And I also owe John a lot because when I arrived in the Corey lab, John was of course already there and was sort of the senior postdoctoral researcher. And in many ways, I also learned a lot from John scientifically. So we will miss John, and I will value his memories as I always have valued George's memories. So let me move on then to talk about science. And here, as many of you know, my laboratory works on two different retroviruses, two different human retroviruses, HTLV1, which I started when I was in George's lab, and HIV1, which I continued after leaving George's lab and joining Malcolm Martin's organization. So when I was asked to speak and give this year's George Corey Lecture, I, for personal reasons and also scientific reasons, elected, of course, to talk about HTLV1 because this is indeed the virus that I started working and the virus that George taught me about. So I thought this would be the more important, more appropriate topic to sort of devote this year's Corey Lecture on. But nevertheless, I want to emphasize to you that in a large part, my career started in HIV sort of using many of the ideas and many of the teachings that I actually learned from George when I was working on HTLV1. So as Michael alluded to, George was of course instrumental in the discovery of the concept of enhancer sequences. And so this was a review article that he and I wrote together all the way back in 1988. The article was actually published the year after George passed away. He passed away on April 25th, 1987. So at the time, George said to me he thought that there would be three ways, mechanistically, that we could explain how enhancers work. So of course, we all understand promoters are the basic nitty-gritty sequences onto which the RNA polymerase cabbages upon and then drives transcription. So enhancers in general are upstream sequences that George thought would, in fact, enhancer would come here, and it would topologically change the promoter, making the promoter perhaps more accessible in a way that was still sort of, at least back then in '88, sort of not well understood. He also thought that looping would be important, where enhancers and promoters would somehow touch each other, even though this would be far away, either in the 5' direction or in the 3' direction, and thereby bring factors to the promoter driving transcription. And the another way that he thought enhancers would work would be that things would bind here, and then they would slide downward, and they would meet the promoter, and therefore drive transcription that would be coming from the promoter. So I think retrospectively now, with the benefit of 3 decades, we know that all of these models are in one way or another applicable to the concept of enhancers. So what George always emphasized to me was, at the end of the day, the key factors that we're going to pay attention to must be DNA binding proteins. DNA binding proteins binding to enhancer, DNA binding proteins binding to promoters, and that is in fact the final effectors that end up driving transcription. So when I left this lab to start my own lab in NIAID, the very first challenge that we had was thinking about this factor that was a transcriptional transactivator. This is the HIV TAT protein that drives transcription from the HIV LTR. And at that time, it was very puzzling because all the lessons I learned from George was that it's supposed to be a DNA binding protein. It is supposed to drive transcription. And it had phenomenological observations that would be consistent with a DNA type enhancer. but none of the experimental evidence fit with that. So one of the things I also learned from George was that when things don't look completely right, you need to pivot your analysis. Okay? And so at the moment when we tried to explain what was going on, I had an epiphany, or I had a new thought that perhaps, in fact, it's not DNA binding, but in fact, although at that time there were no examples of such a thing, that in fact it was a transcriptional activator that bound to a nascent RNA that was driven from the basal promoter. So in fact, that particular notion turned out to be correct, and we published a paper in Cell which demonstrated TAT as the first example of an RNA binding protein that binds to this small hairpin RNA, and in this way works like a DNA enhancer, but is not a DNA enhancer, but is in fact a DNA enhancer-like property that binds to RNA, and represent at that time the first example of transcriptional activation through RNA binding leading to enhanced transcription from the promoter. And so that opened up to me an interest in cloning RNA binding proteins, which we then a couple of years later ended up cloning, I think, one of the first RNA binding proteins in human cells that bound to small RNA. And we coined the name for that RNA binding protein TRBP, for TAR RNA binding protein. Now, some of you younger people in the audience will, of course, have absolutely no clue that this, in fact, is what was the origin of TAR RNA binding protein. Because when I say TRBP to you, you would, of course, immediately say, of course, TRBP. That is, in fact, the critical factor with dicer for the processing of small microRNAs. And you would, of course, be right. But in fact, of course, TRBP was originally identified as a TAR RNA binding protein. And the reason it works as a microRNA processing factor is because, in fact, if you in fact look at TAR RNA, now with the benefit of hindsight 2020, TAR RNA looks exactly like a small microRNA. So those were the sort of the teachings that I appreciate from George in terms of looking at transcription and looking at ways of thinking to pivot your notions if in fact the experimental evidence suggests to you that the phenomenology is similar, but the mechanism might be indeed diverse. Now, the reason I want to talk about HTLV1 is because it was a shared interest between John Brady, George Corey, and I. And in fact, these two papers that were published back-to-back in consecutive years in Journal of Virology in 1987 and 1988 were instrumental, I believe, at the time in terms of defining how the cousin of the HIV TAP protein, which is the HTLV1 TAX protein, works in terms of the motif that it was recognizing and in terms of the pathway in which it was using for activating transcription. So the motif turns out to be 321 base pair repeats, sort of reminiscent of George's 72 base pair repeats in the SV40 enhancer. So these are DNA binding motifs, and the signaling pathway that the TAX protein activates transcription is through the cyclic AMP signaling pathway. And so these are papers on which John and I and George and others collaborated on in those early days, which I think really paved the initial roadmap in terms of our understanding of the beginning of the HTLV1 life cycle, which of course, as the virus enters and becomes a provirus, is at the level of transcription. Now, I know many of you are very familiar with HIV, and I know probably most of you are not familiar at all with HTLV1. So I think one of the things that I want to do then, before I tell you the short stories that I'm going to tell you about HTLV1, is to give you a little bit of background about this particular virus and the disease that it causes. So human T-cell leukemia virus type 1 was the first human retrovirus, and that was discovered by Bob Gallo. And the virus is the causative agent of a T-cell leukemia called adult T-cell leukemia. The interesting natural history of the virus is that the virus infects a human, and then 20 to 30 years later, you have these white blood cells that end up becoming leukemic cells. So there is a very long latency period, so suggesting that there must be multi-step complicated processes after viral infection before that infected CD4 positive T cell ends up becoming a leukemic cell. The virus is also responsible for autoimmune diseases, HTLV-associated myelopathy, tropical spastic paraparesis. It infects about 10 to 20 individuals worldwide, and so it is not by any means an orphan disease, because in reality, the number of individuals infected with HIV at any given time is around 30 to 35 million individuals worldwide. So essentially, the number of carriers of HTLV-1 is about 60% of the number of individuals infected with HIV. Now, the reason why it seems to be very, very much less visible in the United States than elsewhere in places like Japan and South America and parts of Africa is simply because while Japan has a significant prevalence, so does Iran, so does part of Africa and South America, the prevalence of HTLV is really minimal in the continental United States. So very, very few of you would ever hear of anybody who is infected with HTLV or in fact have adult T-cell leukemia as a downstream sequelae of HTLV-1 infection. And I think that is the reason why the disease is not really very visible and well known in the United States, although I think it is a wonderful model in terms of our attempts at understanding an initial stimulus, an inciting event, and how it leads ultimately to transformation. So if you are a physician, chances are you will end up seeing on a very rare basis, and these are slides provided to me by a Brazilian physician who sees, as I showed you on that map, a very high concentration of HTLV-1 infected individuals in Brazil. And people tend to come in with these cutaneous dermal manifestations because originally, as many of you may remember, the virus that was isolated by Bob Gallo was originally diagnosed as a person who had cutaneous T-cell lymphoma. So the T cells, once they become transformed, as Tom Waldman certainly can explain much better than I can, ends up sort of extravasating into the dermal space and presenting with these rash-like lesions. But nevertheless, I think the bad news for many of these individuals is that this is an aggressive T-cell leukemia, and it tends to be intractable and untreatable, and so the outcome is very poor, therefore giving additional impetus to why we should do more to understand the genesis of this leukemia. Now, for the purposes of the molecular biology of HTLV, which is what we spend our time studying, this is a genome organization. Looks very much like a prototypic animal retrovirus with GAG, pol, and envelope, and accessory proteins, of which the key factor that I have spent sort of my career studying is this 40 kilodalton protein called TAX. Now, what happens when a CD4 positive T lymphocyte, normal T lymphocyte, becomes infected with HTLV-1, as I mentioned to you, after a remarkably lengthy latency period, the virus undergoes transformation and gives a very pathognomonic picture, which if you take any sort of pathology board exam, and if you see this kind of flower configuration shape, the nucleus, this is inevitably going to be ATL and not any other kind of leukemia. So when I approached the question then about 2 decades ago in terms of how and why mechanistically does this nice round-shaped nucleus becomes this damaged and distorted nucleus, I thought to myself that the pathology and the histology is telling us something. It's telling us that the disease must have something to do with whole-scale damage to the constituents of the nucleus, which of course is DNA. So there must be wholesale DNA damage, or there must be wholesale chromosomal changes in order for such distortion from this nice, well-rounded shape to this very, very contorted flower-like configuration of a nucleus. So I thought, amongst many hypotheses that one could arrive at in terms of investigating how and why you go from normalcy to aberrancy, that one of the likely and plausible hypothesis would be to investigate the question of how you go from virus infecting the cell, generating distorted nuclear morphology, which probably should be accompanied by gross aneuploidy, markedly gross aneuploidy. And in fact, I think generally that would not be a very bad guess, because in fact, if you look at wholesale series of cancers, all comers, 70 to 80% of them are linked to the aneuploidy of their genomes. By comparison, the numerical linkage is of p53 mutations is somewhat north of 50%. And of course, one of the striking correlations is that if you look at ATL cells, all of them are aneuploid. So that's 100% of them. So suggesting that, as I think many oncologists and cancer biologists coming around, which is that aneuploidy could potentially be the causal reason for oncogenesis, as opposed to the argument which has been floating around for many years, which is in fact aneuploidy is perhaps the consequence of transformation and not necessarily the cause of transformation. So if one were then to accept the hypothesis that aneuploidy is a worthwhile notion to explore for HTLV genesis of ATL, then how does one go about mechanistically trying to understand how that arises? If you think about the genesis of aneuploidy, okay, basically something bad has to happen in mitosis, because during mitosis is when the 46 chromosomes get duplicated and when they are supposed to separate with numerical fidelity, so that when the 46 chromosomes all become double, the spindle pole catches them and has equal segregation, so the one mother cell that exits mitosis ends up with two daughter cells, both of which maintains euploidy. Okay? Now, those euploidies occur because there are two poles. Okay? You can say this is the west pole and this is the east pole, and they throw up ropes tied to the kinetochores of the chromosomes, and then there's equal tension and equal pulling, so then you get equal fidelity of segregation. But what happens, and this happens quite frequently in cancer cells, which is that the bodies that become spindle poles sometimes misduplicate. So instead of two spindle poles, you could have 3 spindle poles, or you could have 4 spindle poles. And in that case, then it would be an unequal tug of war, because when extra ropes are thrown from these 3 spindle poles and they have to be matched against the other 1 spindle pole, you can imagine how 3 poles pulling would pull more chromosomes into one cell than 1 pole pulling in the opposite direction. And so thereby you have the genesis and the outcome of simple physics, where more force goes in this way, less force going this way, more force going this way, and so you have more chromosome going to one daughter cell and fewer chromosomes going to the other daughter cell, therefore the generation of aneuploid cells. And we think then that the generation of aneuploid cells is then one of the early manifestations of ATL, and the accumulation over time is how it becomes a frank leukemic cell down the road. So if that is the case, and if we were to understand that the TAX protein is the oncogenic factor for ATL, then how does TAX go around causing these aberrant numbers of numerical separation? So the answer turns out to be that one of the findings that we came up with was, if you in fact make a fluorescent TAX protein, what you find is that when you introduce it into cells, that every time you see the constituent protein of the spindle pole or the centrosome, the pericentrin protein, that every time you're staying for pericentrin, you invariably find, when you introduce the TAX protein in, that the green TAX protein always goes wherever the pole is supposed to be. So suggesting then that when you have normal mitosis, which would be 2 spindle poles, you would find 2 brightly green TAX-stained polar bodies. If you find 3, this is an aberrant mitosis because it's a tripolar mitosis as opposed to bipolar, and this kind of mitosis will always lead to an aneuploid cell. Then you find what happens is there are 3 green spots. So suggesting that everywhere when you see a spindle pole body, that is the actual anchors that are responsible for fidelity of separation, you will find that the TAX protein there. And in fact, what we think is going on is that the TAX protein actually associates with paracentrum proteins early and cause those paracentrum proteins, okay, by coalescing on them, to misfragment, so that at an increased rate, they tend to fragment instead of into two parts, into multiple fragments, okay, and therefore in TAX expressing cells, you have a much greater propensity for multipolar mitosis, and that in itself becomes a physical explanation for why you will end up with the inciting driving forces for the creation of aneuploidy. So this particular explanation then is not a bad one, because it suggests to us that in fact this is going on. But that can't be the whole answer, because whenever this tries to go on, okay, or if you have a misattachment of spindles, the microtubules from the spindles, okay, when these events are supposed to be going on, okay, we know that normal cells have a checkpoint that says you may form these things, but the checkpoint overrides you and say you may not exit mitosis and you may not divide. Okay? So what is that checkpoint? That checkpoint, of course, is the mitotic spindle assembly checkpoint. Okay? So there are several checkpoints in the cell cycle, okay? But I tend to think about them as two major checkpoints. There is one major checkpoint that's governed by one of George's favorite proteins because it's inactivated by SV40 large T antigen, and the protein that Arnie Levine works on, which is P53. And so that's the checkpoint for structural DNA damage as the DNA goes from G1 to S to become replicated, to be copied. So structural damages must be cleared up before P53 will allow transition from G1 to S. Here now, the structural game is over. What is important here is the numerical game, because when you exit mitosis, you are supposed to have fidelity of numerical separation. The structural monitoring is done here, numerical monitoring is done here, and if in fact, even if you had these unequal tug of wars, okay, they are not supposed to exit mitosis as long as the spindle assembly checkpoint is in fact intact. So we knew that in TAX expressing cells in ATLs, multipolar mitosis occur, okay, and multipolar mitosis are allowed to, in fact, consummate to produce aneuploid cells. So suggesting to us then something else must be happening to this checkpoint, even though we had only identified the TAX protein as associating with the spindle pole bodies. So in order to answer this question then, a postdoctoral fellow who joined the lab decided that this must be a nuclear interaction occurring during mitosis. So he went ahead and he cloned every single TAX binding protein in the mitotic phase of the cell cycle that bound to the TAX protein. And from that cloning, we identify the protein that was at that time postulated to exist from yeast mitotic experiments as the mitotic arrest deficiency protein number 1. So this turns out to be the mitotic spindle assembly checkpoint protein that the TAX protein also has to bind to and also has to end up inactivating. So it turns out that there are two different events that's going on. One is the event that causes the physical reason for generation of aneuploidy. And then there is a second event, which is that the TAX protein has to inactivate the sentry, the policeman that says, when something bad happens, you are still allowed to live, okay? And you must not be sent into apoptosis and be committed to die. So by doing two different things, creating the physical reason for aneuploidy and also inactivating the checkpoint that censors against aneuploidy, we believe that the TAX protein allows the consummation of the formation of aneuploid cells, which are in fact the leading events, the early events for the genesis of ATL leukemia. Now, when we published this paper in Cell, there was a lot of brouhaha. And people said, you know, this paper really doesn't answer the question of spindle assembly checkpoint and cancer. Because all it did really say was the MAD1 protein binds to an overexpressed oncoprotein. And we really don't know whether the oncoprotein is simply only inactivating the MAD1 protein to cause transformation and to cause these proliferation defects, or when you overexpress the oncoprotein, there are lots of things that you don't choose to see. Of course, you won't see them. They are actually responsible for the changes. Okay? And so the findings here are, in fact, not a fait accompli in terms of demonstrating that this checkpoint and this particular protein has any role in terms of transformation. So we sort of fretted over that for a while, and then we finally decided that the simplest way, of course, to answer that question was simply to make the knockout, right? So if you knock out this particular checkpoint protein, as many other checkpoint proteins have been done with, if the checkpoint protein is important for guarding against tumorigenesis, then removing the checkpoint protein should be, in a very simple way, show up as increasing tumorigenesis in the knockout animal. And that experiment took us a couple of years to do because we were just learning how to do knockout experiments. And in fact, we ended up creating heterozygous knockouts of MAD1. The homozygous knockouts turned out to be embryonically lethal. But nevertheless, if it's an important checkpoint and you lose only 50% of the function of that checkpoint, it should still be sufficiently clear to manifest as an increase in the number of tumors. And in fact, it did turn out to be the case. So as you can see, in a fairly large series of mice, so over 100 mice, wild-type mice, we all know that wild-type mice has a basal level of tumorigenesis, spontaneous tumorigenesis upon aging. And so when you age these mice, by the time you reach about 2 years, spontaneous tumorigenesis is about 18 out of 130, and the heterozygous knockout of this checkpoint protein ends up showing up a doubling of the rate with very significant p-values of 0.0043. So suggesting, in fact, that our particular hypothesis is correct. Inactivation of this particular checkpoint leads to increased aneuploidy, and inactivation by the HTLV1 oncoprotein creates that, generates aneuploidy, and aneuploidy ultimately causes tumorigenesis. Now we can embellish upon that particular experiment in the following fashion. We know now, although in the early days we knew less, but we know now that in fact MAD1 is only one of several components of the spindle assembly checkpoint. Many of these are somewhat redundant, although many of them are also independently critical to the checkpoint. So we know that in fact the MAD1 and the MAD2 proteins are heterodimeric partners. And so we, of course, made a weak non-functioning checkpoint because we could only make the heterozygous animal. So we thought to ourselves that perhaps the experiment could be reinforced, that the notion and the hypothesis could be reinforced. In fact, if we could make compound animals with the knockout of also the MAD2 protein and the MAD1 protein, and then marry these mice together to make babies that would in fact be doubly knocked out for MAD1 and MAD2. Now each homozygously knocked out is lethal, so we could then of course only make compound heterozygous knockouts, MAD1 hetero knockout, MAD2 hetero knockout, and we generated a number of independent mouse embryo fibroblast clones that are wild-type MAFs. MAFs that are only heterozygously knocked out for one checkpoint protein, MAFs that are heterozygously knocked out for the second checkpoint protein, and MAFs that are doubly heterozygous knocked out for both of the checkpoint proteins. And so the prediction, and the cell biology verifies the prediction, that there is an increased propensity towards aneuploidy as you go from wild type to these doubly knockout. And in fact, knockout of this particular protein is more powerful in inactivating the checkpoint than knockout of MAD1. MAD2 is more critical than MAD1. MAD1 tends to be a regulatory partner of MAD2. And the bottom line is that as you go increasing in the degree of propensity towards aneuploidy, you increase toward the degree of tumorigenesis, okay? So wild-type MEFs don't make any tumors when you inject them into mice. MAD1 heterozygous MEFs are more tumor-prone. MAD2 are even more, and the double heterozygous knockouts become even stronger, okay? And so this experiment, we show you some examples here, but it has been replicated and indicating that, in fact, the whole idea is, in fact, true, okay? That the more you generate in terms of cells that tend to become aneuploid, the more potent they are in terms of tumorigenesis when introduced back into animals. So then you say to yourself, but at the end of the day, these are still mice, right? What is the evidence for MAD1 inactivation in human cancers? And in fact, if you look very hard and you look for genetic mutations of MAD1, okay, you don't find them very frequently. So a lot of people have argued that these are interesting and fine observations, okay, in mouse systems, but perhaps they don't have any real relevance to human systems. It turns out that that's not correct, okay? And that's not correct because, in fact, several years ago when we surveyed a whole series of colon cancers, what we found was that the genetic evidence is correct. Inactivation of MAD1 genetically, in terms of mutation, is in fact very rare. But what we found was over 80% of the colon cancers, and these are primary colon cancer samples, in fact have lost mitotic spindle assembly checkpoint function by overexpressing the MAD1 protein. So at the time when we reported that, people sort of said, well, no, I mean, you know, that's just sort of their rationalization because they can't find MAD1 genetic mutations. So it turns out that a couple of months ago, as I was reading through, catching up on the MAD1 literature, this particular paper was published in the Proceedings of the National Academy of Science. Basically, and it's always gratifying when people in their abstract cite your paper outright at the beginning and say, oh, based on what they found 5 years ago, we in fact went and looked. And in this case, they looked in human breast cancers, and they found that over 70% of human breast cancers behaved like what we had seen with colon cancers. All of them, all 70% of those, are inactivated from mitotic spindle assembly checkpoint because by virtue of overexpressing of the MAD1 protein at the protein level as opposed to genetic mutation of the MAD1 gene in terms of inactivating that particular protein. So overexpression as well as loss of expression both end up inactivating the spindle assembly checkpoint. And the key message is then that inactivation of the spindle assembly checkpoint, whether it be by genetic mutation or by overexpression, in the mouse case, we can demonstrate it by genetic mutation. But in human case, apparently it is occurring by overexpression of the MAD1 protein, leads to the same final outcome, which is cancer oncogenesis. Now, in the first part, I talked about HTLV1 and aneuploidy. I want to switch gears a little bit and talk about a shorter story, which is about HTLV1 and serendipity. Okay. I mean, one of the favorite questions that I'm always asked by many of my colleagues outside of NIH is that what is the benefit of you guys working at the NIH? You know, other than the fact that you have to fill in all sorts of ethics forms and you don't get approved to travel without ethics sort of going through with a fine-tooth comb. And I always say that one of the benefits that we have as community researchers at NIH is that we get to be able to follow serendipitous findings. And a lot of times that ends up being some of our best science. So part of our serendipitous finding that I'm going to tell you, then this is a much more recent story, which is that, and it follows from our study of the MAD1 protein, was that we wanted to understand how this particular protein ends up from, of course, translation in the cytoplasm, and at some point having to go into the nucleus and going to the spindle pole bodies and sitting on kinetochores, okay? How does it go? What is the journey of this particular protein? And could we learn something from where it stops and what partners it holds hands with. So by and by, as we were studying the translated protein and then as it moves into the nucleus, poised when mitosis begins to go to the kinetochores and to the spindle pole bodies, we found by, at that time, a crude proteomic analysis that the protein was in fact binding to an inner nuclear envelope protein called SUN1. Okay? And so I said to the postdoc, I said, well, you know, here is a simple experiment to do because now we've gotten much better at making knockout mice. What we should do is that we should, you know, this is a regulatory protein of the MAD1 protein. Okay? MAD1 has to come and sit here and bind to this inner nuclear envelope protein before it goes to doing its mitotic spindle assembly checkpoint function. Okay? We knock this guy out, the whole spindle assembly checkpoint falls apart. Okay? MAD1 knockout is homozygously lethal. Maybe this one is not, so this gives us a chance to answer this question in a clean fashion. So the postdoc agreed, and we made the knockout of this particular protein, and lo and behold, we were very excited because this one we could knock out and we could breed to homozygosity. Okay, so we said, okay, now the waylay station for MAD1 on its way to kinetochore to do its sentry function, okay, is now destroyed. So the checkpoint must become inactivated. Okay? So therefore, all we have to do is sit here and look at these animals, look at these mice. Okay? They are born alive, and so we can follow their phenotype, and let's just wait for tumors to form. And we waited and we waited and we waited, and everything was normal. No tumors. Okay? And so the postdoc, you know, worked very, very hard, okay, because you spend a lot of time, you know, developing a knockout mouse, and you really need to get some insight into it. And so she found that, in fact, nothing was very remarkable except that these animals have problems with gametogenesis. Okay? So in this case, sperms don't form very well. And of course, gamete-related small RNAs, which we were also interested, piRNAs didn't form. And so we were able to publish a paper describing these findings, but it was still rather unsatisfying because our original model was that this was supposed to lead to some sort of disease manifestation, and we were really interested in finding a disease phenotype. So around this time, I was sort of fretting about this, and I said, this is an inner nuclear envelope protein. So maybe what I should do is I should read up a little bit about what nuclear envelope proteins, when they are not working, what sort of disease they're supposed to cause, because again, it's a new area for me. So I came across this review paper at that time, which was published by Colin Stewart, and Colin, as many of you know, used to be at NCI Frederick before moving out now to Singapore. And so Colin is, of course, an expert in nuclear envelope proteins, and he wrote this very, very illuminating article basically describing that a number of human genes and proteins that are responsible when they have been lost for function, they are responsible for nuclear envelope distortions and aberrations. And those diseases turned out to be muscular dystrophic type of diseases, okay? Of which one of the more important category, okay, are the lamin A mutation and the Emerin mutation, responsible for diseases like Emery-Dreifuss muscular dystrophy, and of course lamin A is responsible for this very famous disease of which Dr. Francis Collins is the world-renowned expert on, which is Huntington-Gilford progeria. Okay? So I said, I don't understand. Everybody else's nuclear envelope gene knockout ends up developing into these very, very dramatic phenotypes. And our nuclear envelope knockout, and clearly the SUN1 protein, is one of the major components of the nuclear envelope, okay, in the nuclear envelope. And ours just sort of, you know, the mice just sort of look back and blink at you, and there's really no phenotype, except the mice don't like to reproduce children, okay, and they are infertile. So I said, what can we do about this? And so sitting down discussing with the postdoc, I thought, well, perhaps what this is that this is not a major factor, but maybe it's a cofactor. Okay, so we know that there are mouse models from developed by Colin Stewart that are knocked out of the lamin A, and those tend to replicate what people see in Huntington-Gilford progeria syndrome, okay, which is that they have a rapid aging phenotype and they all die prematurely. Okay? This mouse, for example, knocked out the lamin A, dies within 50 days after birth. Okay? That is extremely premature aging. Okay? So if we think that this guy does nuclear damage and nuclear envelope distortion, if we marry this mouse to our SUN1 knockout mouse, would we not end up with a faster aging phenotype and an even more dramatic nuclear distortion? And we would end up developing a faster aging model. And in fact, that would be the cat's meow. And people interested in aging would then want to come and ask us for our mouse model. So I suggested that to the postdoctoral fellow, and she again was very acquiescent about it and ended up creating the compound married mouse, in which we thought would end up into an even more lethal, faster aging phenotype. And lo and behold, what we found was, as you recall, I told you this particular single knockout results in 100% lethality by day 50, although all the mice are born normal. Now, interestingly, and this is something that tells me that my mother, and your mother probably told you the same thing, isn't always right. Because if you remember, Mom used to always say, you know, T, two wrongs don't make a right. So here is one wrong. Now here we create two wrongs. And now, in fact, we rescue. Instead of greater lethality, we end up rescuing the mice now back to normal aging. Almost normal aging. This is normal aging. This is normal aging. But this is pretty darn good because the p-value is down to 0.00001. Okay? So this tells you that something is going on. Okay? And this tells you that in this mouse lies a clue to why this mouse ages prematurely, because this mouse ends up curing the aging phenotype that is coming out of this mouse. Okay? So what is the answer? So we end up recently publishing this paper in Cell, and the answer turns out that the interesting outcome of one knockout, which is a knockout of the lamin A, is that the lamin A knockout itself, we think, is not directly pathogenic. But what happens is that when you lose the lamin A protein, the nuclear envelope is not synthesized properly, and the cell seems to think that the reason it's not synthesizing the nuclear envelope properly is because it's not making enough SUN1 protein. So it keeps on churning out the SUN1 protein as a compensation for the loss of the lamin A. So what you find in lamin A MEFs is you have gobs and gobs of overaccumulation of SUN1. And of course, it makes sense when you knock out the SUN1, the lamin A loss in itself is not pathogenic. When you knock out SUN1, it can't over-churn out the SUN1 protein. So therefore, the direct reason for pathogenesis, which is overexpression and inability to rid of this overaccumulated moiety, is now solved, okay? And therefore, aging comes back to normal. So is this in fact the same with the human example of lamin A mutation, which is the Hutchinson-Gilford progeria syndrome? And in fact, it turns out, at least from what we've seen, to also be the case. Because in HGPS, there are also cells which overexpress SUN1, okay? And only the cells that overexpress SUN1. Some cells, so these are normal cells, they dimly stain for SUN1. And in HGPS, 1 out of every 3 cells is overexpressed for SUN1. But only these cells that are overexpressed for SUN1 in HGPS are distorted in terms of nuclear morphology and are actually pathological. The ones that are not overexpressed for SUN1, even though every one of them have the lamin A mutation, some of them just don't turn out to be overexpressed for SUN1. Some of them overcompensate for overexpression of SUN1. And the ones that overcompensate for overexpression of SUN1 resort in these distorted, crenated nuclei and end up being the pathological cells. And so if one is able to use siRNA to knock them down, then what you find is that the cells that are not knocked down remain crenated. But all the cells in HGPS that are knocked down for someone now recover their normal nuclear morphology. And we predict that those cells would in fact be physiologically normal, okay? And that it's the overexpression that ends up being the bad actor. So what we think is happening, okay, in dystrophic diseases, okay, at least from the example that we've worked on, is that when you have a mutation in one entity, there is compensatory oversynthesis of proteins of other entities, okay? And in this case, when you have a mutation in lamin A, you have oversynthesis of the SUN1 protein, and it's the oversynthesis per se, without getting rid of them, that ends up distorting the nucleus and creating the pathology. So therefore, it is fully in keeping with a paper that was recently published in Science Translational Medicine from Dr. Collins' lab, which says that if you now increase the protein degradation pathway, which rapamycin is an inducer of the autophagy pathway, which gets rid of these overaccumulated protein aggregates, then treatment with that particular drug is consistent with treating HGPS and returning back to normalcy. And we think abnormal aging and normal aging may very well be mechanistically the same. Because as you know, there is also a body of literature that says if you treat mice, normal mice, with rapamycin, which of course also improves their junk protein degradation pathway, those mice acquire higher increased longevity. So another way of looking at the hypothesis then is that here is the normal cell. This is what happens to the cell that in fact is knocked out for lamin, so SUN1 overaccumulates, and then this is what happens to the cell when you have knockout of the lamin and knockout of SUN1. These overaccumulation goes away, and sort of all these bad things that happen when the nucleus gets distorted, like hyperactive DNA damage and abnormal transcription and cytotoxicity from overaccumulation, they all become resolved, and therefore you return back to normal longevity in most of these mice. So I want to close by saying that, you know, although it's serendipitous, I think in many ways, in fact, when you do good scientific findings, they sort of end up converging and dovetailing together, which is that we also know that aging also results in weakening of the checkpoint, and aging also results in increasing aneuploidy. And so my guess is that somewhere downstream, when you look at some of these premature aging mice, you will find that they have increased propensity for aneuploidy with those nuclear distortions and with also DNA damage. And my guess is that somewhere, if they lived long enough, we would have found that they would have increased prevalence of tumorigenesis. So let me wrap it up by summing for you that in the first half of my talk, I talked to you basically about HTLV1 transformation. I told you only a little snippet of what our real program has been. So I've told you only about this particular aspect of our research. In fact, there are all these different other areas which all these different postdocs over the years have participated in. All these other steps are also important, like generation of reactive oxygen species, of also activation of microRNAs and so on, that I haven't had time to touch upon. And so I think this is very exciting for us. I'm very grateful to George for sort of starting me on this particular system. And we think we've made good mileage in the road traveling to trying to understand how the virus causes leukemia. And by, I think that the wonderful community of doing science at the NIH is that we are able to pursue interesting leads. And I simply want to sum up the second half of my talk by sort of highlighting this particular News and Views type article that was written in Current Biology when our paper was published. And basically you can sort of cogently sum up the issue here is that, as mother would always tell you, too much sun is really bad for you. And so in our case, too much sun will really make you age faster and certainly do bad things to your physiology. Now, Let me close with this slide to acknowledge current postdoctoral fellows in the lab, previous postdoctoral fellows who've worked in the lab, who've contributed to some of the stories that I've told you today, some of our collaborators, other laboratories, NIAID intramural funding. Of course, over the last 18 years or so, I've received funding from IATAB, and we've also had a challenge grant that was awarded to us from Bill and Melinda Gates Foundation. Now, I want to close by making a remark about three things that George taught me, separate from science, which I would really like to acknowledge. George taught me about three things that are important. He says, always remember your family, your friends, and your community. And so I want to close by acknowledging, of course, this is a family picnic that the society that I was privileged to lead with a chapter at NIH, Society of Chinese Bioscientists in America. This is a family picnic that we organize every year, every summer. This was at the Cabin John. And I think it's a remarkably wonderful occasion for us to sort of recognize the importance of the family in terms of allowing us to do the long hours and sort of the committed work that we do at the NIH. And finally, I want to give a few words of thanks to, so of course, many of you, most of you are my friends sitting in the audience, and I really appreciate that you've come. But within our larger NIH community, as I mentioned, there is a smaller community of Chinese bioscientists who I meet with every month, and in many ways we collaborate and we offer each other advice and support. And so some individuals that I would particularly like to recognize, you all know Paul, you know Tsu Sha, you know Yunbo, you know KG, Jake, Xing, and a number of other individuals here. And this was last year where our society and our chapter organized together with NIAAA a symposium in Building 31. And many of these individuals over the last 27 years have provided really important guidance and support to me for my career at the NIH. And for all of you, for my family, for all the friends, many of you in the audience, and certainly for the greater NIH community, those are the things that George has taught me are important, and I thank you.
Although the hour is a bit late, I think George would not rest easy if we didn't have some questions. Tom, you have a question.
The second half of your lecture is aging as early sunset. I would like to turn to tax and aneuploidy. Through the course of HTLV-1 infection, which for many people is lifelong, that is, it starts at birth, one has a carrier state where tax is expressed. During that period, is there increased aneuploidy? And at the other end of the disease, over half of the patients with acute adult T-cell leukemia no longer express tax. They express HBZ, but no. Can one have this aneuploidy continue in that phase in the absence of tax?
So let me answer the second part first. So we think that in the case of ATL, it is a little bit different from other viral caused tumors. Okay, so for example, you know, I occasionally serve on study sections, on virology study section, and I would have this running argument with HPV virologists, and they would argue that in their case, if you lose E6 and E7, you can never maintain the cervical cancer. In our case, as you know, we lose tax, and the cells are happy to be ATL cells. So I think what happens is that you know, of course, chemical-induced carcinogenesis has no oncogene, no viral oncogene. So it is simply, you know, change in the genetic complement of that particular genome. So I think you reach a certain stage where if you have aneuploidy, you have acquired that all the independent proliferation and anti-apoptosis and anti-senescence signals, okay? So then you no longer need tax, okay? So we think that's the reason why you don't need tax at that stage. Now your first question is, well, in early infection, okay, does aneuploidy occur, and how do we measure that? I have to profess, we don't have evidence, so we can't tell you. But I could sort of confabulate a little bit, okay? And my confabulation would be, there is a struggle when the virus infects the cell, okay? Because, as you know, the first cell of defense, okay, and cells are incredibly more altruistic than you and I are, okay? Cells are willing to commit suicide before, allowing another cell to become infected. So I think the first signal and the first intent of the cell when it's infected by a cell, okay, infected by a virus, would be to commit apoptosis, or in some of Joe's studies, the cell would commit senescence, go to sleep, to protect against spreading the infection. So I think if you, in that situation, express too much tax, you're obviously going to go into apoptosis. So there's a selection for only cells that express, I think, a very modicum amount of the tax protein. And those cells, I think, will not give you whole-scale acquisition of aneuploidy, but slowly you will gain trisomy 21, which is perfectly viable, trisomy 18 is perfectly viable, and that kind. And then the things start rolling over time. And so I think the really dramatic changes, those are eliminated through apoptosis, through senescence, okay? And so you get the mild guys, okay? And they become sort of the nidus from which accumulations occur. And then finally, when you have sufficient accumulation, you become now tax independent. Okay, 90% of that's confabulation. I think 10% is probably rooted in observations. David.
Hi, T. That was a terrific talk. So from the point of view of the virus, because presumably tax evolved this for the viral life cycle, not for the sake of producing tumors, what is the advantage to the virus for the interaction with MAD1?
So I think the advantage for the virus is that the virus, so I think the virus is basically pro-proliferative. And transformation is a perhaps unwelcome but unavoidable side effect of whenever you increase pro-proliferation, because this is a virus that does not produce a burst size of viral particles. So basically the only way that you amplify the viral genetic material is that you make the integrated provirus in the cell that carries the integrated provirus divide more and more and more. Okay, so it's very different from HIV. Doesn't release the burst of viral particles, okay? It simply just basically dribbles out no virus. So I think it behooves the virus to really, I think, the smartest HTLV would be an HTLV that immortalizes T cells without going over the tipping point of making them into cancers. Okay, so historically, as you know, many cancer biologists study, transformation in vitro, and we always think about the first step is immortalization, and then that's followed by transformation. Immortalized cells proliferate ad infinitum, but they cannot cause tumors in vivo. But when you tip over to the transformation stage, then not only can you replicate forever, you can also cause tumors when you inject them into animals, okay? So I think HTLV, unfortunately, has evolved to be a little bit overly robust. The best HTLV would be an immortalizing virus, but not a transforming virus. But I think in the quest for efficient immortalization, the virus has also acquired the ability to transform. Sure.
I'm still trying to square your morphological observation of binding of labeled tags to spindle proteins versus your mechanistic studies. You said in aneuploidy, The spindle proteins are all derived from fragmented kinetophores. Okay? But the tax is nonetheless able to bind to these poles which have arisen out of fragmented starting point or precursors. Does that mean the tax can bind non-specifically to the components of the pole, or that they all the poles which are assembled regardless they're coming from fragmented parts, that they're all competent to bind the tax?
So the simple explanation would be tax is directly able to bind paracentrin protein. Okay, and therefore, every time where you see paracentrin, okay, you will find tax, and which is what the staining tells us. And when tax binds onto paracentrin, the paracentrins tend not to be able to form, the bipolar program. Instead, tends to form multipolar program. That's the simplest explanation.
If MAD1 is the target,
MAD1 is a separate moiety.
So
it's different from paracentrin. Yeah,
they're not connected.
it's a different, different target. Okay.
Okay, well, I think when George started working on SSB40, his thought was that a simple virus would, which had such a profound effect on cell biology, would allow us to learn a lot about how the cell works. And I think George would be extremely pleased to know how far this work has come. T, this was a terrific talk. I want to invite you all to a reception sponsored by the FAS in the library, and hopefully we'll be able to continue the conversation there. Thank you very much.
Thank you.